in order to get the strongest of your immune response. So what you do is that you give a vaccine. Vaccines would be this initial antigen. Antigen is any foreign protein that you're putting in. When you put that in, you get this initial response, but I want you to show you what the secondary response looks like, either for a booster vaccine shot or if you actually encounter the pathogen. Number one, not only do you get a higher response, but it's a much faster response. What took 10 to 14 days over here now takes just a day and a half to 2 days because the system has been primed. You have circulating antibodies that have been produced specifically against that microbe, as well as memory cells that are circulating that when they see this pathogen again, they're able to respond in a short time instead of this long buildup that is necessary. In other words, you have the pathogen and it's in the database of the body and the body can respond quickly. So the reason for this, the reason for actually having this initial break is that it doesn't matter how well you've eaten. It doesn't matter if you've always lived on a vegetarian diet. It doesn't matter if all your vitamins are in place. Nothing at all substitutes for that initial primary immune response. Without that, you're still going to have to wait 2 weeks in order to have that strong response. And that is really important when you're thinking about a vaccine. That's what the vaccine is going to do. It will give you this head start so that when you actually encounter the virus, you already have in your circulation these neutralizing antibodies or these antibodies that can bind the virus up, as well as memory cells that are ready to go to produce more of that entity that you need. And do vaccines work? Of course they do. We've seen that with all sorts of examples of diphtheria and pertussis and measles. You can see how many infections you have by the size of the circles before a vaccine came around. And then you introduce a vaccine here, there, and there, and the numbers go down dramatically. Now, what is the goal of vaccination? It's not necessarily to vaccinate every person, but it's to vaccinate enough people to get what we call herd immunity. And herd immunity is when you have so many people who have been protected in the community that those who are unprotected have a very little chance that they'll actually get infected. And so in this case, here you have a few people who have been vaccinated in blue in this diagram. And then you have one person
sharing the audio.
You have one person who is infected. And this one infected person can now go and infect others.
Yeah, I mean, as long as he knows it's still being recorded, that's...
Stelendor, we can hear you. Please mute. You can have all of these people now getting infected. When you have the opposite scenario, where you actually have most people vaccinated, 80% or thereabout people vaccinated, one person is infected, the probability of that person infecting many people goes down significantly. That's the whole idea of herd immunity. For COVID-19, the thought is that you need to vaccinate about 75 to 85% of the population to reach to the stage of herd immunity. Because remember, there are people in the population who cannot get a vaccine. They have underlying conditions that prevent them from getting a vaccine. Now, this picture that I'm showing you is kind of hard to look at because this 13-year-old on the left actually has smallpox. But what you should know is that these boys got exposed to the disease on the same day from the same person. And the only difference is that the one on the right got vaccinated and the one on the left did not. Vaccines work. Here is a line in the at Mrna Hospital in 1947, where people are lining up to get vaccinated against smallpox because it's such a devastating disease. It affected so many people, killed so many people.
That was polio. Yeah.
Everybody.
You know how roses are red became? Roses are red. No.
Just needed to have that vaccination happening. So again, the idea is here's your spike protein of SARS-CoV-2 binding to the receptor here. And the idea is that if we have neutralizing antibodies, those antibodies can come in and block, because you see this spike protein is like a key that the virus uses to unlock our cells in order to get in. And so what you're doing, you're literally coming and messing up the key, blocking the site that the key would attach to get into the human body. That's the process of a vaccine. Now, there are many, many vaccines that are being developed against SARS-CoV-2. There's never been an infection that has had such a huge effort to try and develop vaccines. So there are 37 in phase 1 where they're just looking at safety, 24 in phase 2 where they're looking at safety and efficacy, and then phase 3 is a trial where you actually look to see how well the vaccine works while you're still looking at safety. So we have 2 vaccines approved here in the United States, another 8 vaccines that have approval in different parts of the world, including the Astrazeneca vaccine that has approval in the UK. And there are 4 vaccines that have been abandoned for various reasons at this stage. And you're abandoning a vaccine, especially after you've gone through phase 1, 2, or even phase 3 trials, costs lots and lots of money. So here's a listing then of the vaccines that we have. Moderna and the Pfizer BioNTech vaccine have been approved in the US. The Astrazeneca vaccine has been approved in the UK. You have the Gamaleya vaccine that's been approved in Russia. The Johnson & Johnson vaccine, they just got phase 3 clinical trial data out. There's about a 60% efficacy, which, by the way, is still really good. We've been spoiled with these 95% efficacy vaccines, but it's a real good vaccine, and it's a single-shot vaccine, the J&J vaccine, and that should be going up for approval for emergency use authorization really, really soon. So this is the vaccine landscape. Now, I just want to show you a little clip on the human genome. The human genome was sequenced in 2000. Where they were able to see all 3 billion letters that make up the human genome. And this will give you a little idea of what DNA is, what RNA is, so that in the next slide, when I talk about RNA vaccine, we will be on the same page. So just listen to this little.
A sunny day, a simple friend, and a complex biological story. The Human Genome Project is a way of exploring our molecular selves. Almost all of our cells, the muscle cells that let us smile, the brain cells that perceive the humor in things, the cells of our eyes that take it all in, contain a complete set of all our genes. The genome. If we could journey inside ourselves into a cell, we would see 23 pairs of chromosomes packed into a nucleus. Each chromosome contains a long coil of DNA. If all the chromosomes were unwound, the DNA in just one of our cells would stretch 6 feet long. The DNA double helix contains 4 kinds of building blocks. An A always pairs with a T, a C with a G. DNA contains information to make every part of our bodies with its 4-letter language. Each of our thousands of genes codes for a specific part. RNA polymerase copies the information in a gene into a messenger molecule, messenger RNA. The building blocks of messenger RNA and DNA are called bases. The bases on one strand of the DNA specify the order of bases on the new strand of messenger RNA. The DNA always stays inside the nucleus, but messenger RNA travels out into the cytoplasm. There, a protein-making machine called a ribosome can read messenger RNA to make a particular protein. Every 3 bases of the messenger RNA molecule codes for an amino acid. Proteins are made of amino acids. tRNA molecules help translate the language of DNA and RNA into the language of proteins. tRNA molecules bring the right amino acids that the ribosome links together to make a protein. Proteins are the laborers. Some form structures like tendons and hair. Others perceive light, scents, and flavors, control chemical reactions, and carry messages between cells. To understand our molecular selves, scientists have read the 3 billion letters making up the DNA in the human genome. Different sets of genes interacting with complex environmental factors influence things like our looks, personalities, and risks for diseases like cancer and heart disease. A growing understanding of our genes and all they do will help us understand the complexity and the wonder of life. A simple friendship, a sunny day, a life.
And this essentially is what the psalmist was talking about when he said, we're fearfully and wonderfully made. With these 4 letters of the DNA alphabet, you have the coding for every single thing that is made in our bodies. And this is in every cell of the body. Now, this is a cartoon of a cell. The purple thing in the middle is depicting the nucleus. The DNA that you just heard about lives in the nucleus, right? That's where it stays all the time. The proteins that they just talked about are made in the cytoplasm. So the cell has 2 compartments. This outer area here where all these organelles are is called the cytoplasm. The inner part here, the purple part, is the nucleus. Right? Really important. The DNA resides in the nucleus. In fact, the cell thinks that the DNA is so very important that it doesn't allow it to leave the nucleus. Stays in the nucleus. And so then how does it make proteins from that instruction in here? It uses the messenger RNA. Okay. So the way I've described it before is, let's say that you have this great book. Maybe it was written 150 years ago with these unique recipes in it for certain pies and cakes and other types of things that you can make in the kitchen. And you want to make this special pie, but the only way to get the recipe is actually to go to the library and get this very old book that they're in. Now, the book is so unique that the library won't allow you to leave the library with it. It's on reserve. And so you have to go now pull it off reserve and photocopy the pages that you need, the pages with the recipe on it. And you take that now to your kitchen where you read off your recipe, you grab the ingredients and you make the cake. The DNA is depicted by the book, this unique book that's in the library. And the library is the nucleus. Your kitchen would be the ribosome. This is where proteins are made. And the photocopy would be the messenger RNA that you take out. So that's what that messenger RNA is. It's the instructions for how to make the cake. So you go in your kitchen and you make the cake. So there is what we call the central dogma of molecular biology, that you go from DNA to RNA to proteins, or you do what is called transcription, translation. You get a transcript of a particular gene that is in the nucleus. That transcript, that messenger RNA, leaves the nucleus, comes out into the cytoplasm as messenger RNA, and is translated the same way you're translating whatever ingredients are on the paper into making stuff, a product in your mixer, and then putting it in the oven. And that's what a protein is. And so here's an animation that shows you a lot of processing happens of the messenger RNA. And I'm spending time on this because it's important to understand the biology, even a little bit, if you're going to appreciate how the vaccine is made.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds, the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay, so this is like a factory. We have inside of our bodies this protein making factory, and it will make any protein whose code it gets. And so essentially then, what these companies, Pfizer, BioNTech, and Moderna have done is that they've utilized this knowledge, because you see, when the virus infects us and it gets inside of our bodies, the virus literally instructs our bodies to make the proteins that it needs to reassemble itself. So what these companies have done is that they've taken the messenger RNA for just that spike protein that the virus uses to attach to our cells. And it has encapsulated in these lipid nanoparticles. So these are just fat molecules. They found out some time ago that if you encapsulate it in fat, it protects that messenger RNA when you inject it into the person. And now you can actually get it inside of the cells. Once it gets into the cell, it does not go into the nucleus. It stays in the cytoplasm. That is where the factory is. So you have to think about the cell as being 2 rooms that are round rooms. One inner room, the nucleus, that's where the DNA is. The outer room, the cytoplasm, it has several other things, but it has that protein factory there. And so what Pfizer, BioNTech, and Moderna did is that once the sequence of this virus came out in January, they didn't need to do anything but sit at their computers, look at what this genetic code was, and then they figured out what the exact code for the gene that make that spike protein, that key that the virus uses to unlock our cells. They literally took it over now to a piece of equipment called a DNA printer, where it makes a DNA copy, and then synthetic messenger RNA, in other words, in the lab, messenger RNA is created, packaged in these lipid particles that I talked about. And that's what's in the vaccine. It gets injected in you. And once it gets into the system, your body now takes these messenger RNA copies, these photocopies, and they read off the ingredients that it codes for and make proteins, the spike protein. So if you see, these little spikes are being made, not the virus itself. So here's an animation of that.
mRNA technology has never been used before in an approved vaccine, but scientists are using it now to combat COVID-19. mRNA, or messenger ribonucleic acid, are snippets of genetic code that tell cells to build proteins. mRNA vaccines take advantage of that protein-building process to trigger an immune response and build immunity to SARS-CoV-2, the virus that causes COVID-19. Instead of using a small dose of live or dead virus like some other vaccines, mRNA vaccines use a tiny piece of the virus's own genetic material. Here's how it works. This is the virus. These are called spike proteins. Scientists have isolated the genetic code for those proteins, a set of instructions that are put into the vaccine so your body knows how to make them. Your immune system won't make an actual virus particle, but it will make copies of the angry red spike protein. In other words, no gray ball, but an angry red spike. Once vaccinated, your body starts making those spike proteins so your immune system will develop antibodies to fight the real thing, SARS-CoV-2, the virus that causes COVID-19. The Pfizer and Moderna vaccines that are created for COVID-19 will be the first licensed mRNA vaccines ever.
So it's important that you realize that although these are the first mRNA vaccines that will ever be created, the mRNA technology is not new. Here's a paper that was published in 1992, February of 1992, that actually was looking at the mRNA technology to reverse a type of diabetes, right? So the whole idea of mRNA as a therapeutic was there. Here's a 2017 paper looking at using mRNA to make a rabies vaccine. Here is another paper that was published in 2019 looking at making a flu vaccine using mRNA technology. Here is a release from Moderna looking to make vaccines using this mRNA technology for Zika virus. Here is another paper, January of 2020. January 9th of 2020. At that time, the sequence had just come out. They're putting out a news release here that they're trying to make a cytomegalovirus vaccine, and they're in phase 1 trial. In other words, they're already in humans trying to make a vaccine against this. And I could go on and on. mRNA vaccines, a new era. This is a 2018 paper that was published. Here is another one looking to use it for type 2 diabetes, 2019 paper. And so again, this is not a new technology. It's just that we've never had a vaccine go to market before using the technology. But the technology is about 20 years old proper. So what is in these vaccines? What is actually in these vaccines is the mRNA that I talked about, multiple copies of it, plus fats, because it's encapsulated in this fat ball. You can think about it as a little ball with the messenger RNA inside of the middle of it, and this fat is around it. There's polyethylene glycol, which is used to actually hold the membrane together and help it to fuse to the cellular membrane, and then salt and sugar. These vaccines, the Pfizer, BioNTech, and Moderna vaccines have no preservatives in them, right? And pretty much all they have is the messenger RNA, the fat, and all of that. Neil deGrasse Tyson, that many of you might know, one of my favorite scientists, said, one of the greatest challenges in this world is knowing enough about a subject to think you're right, but not enough about the subject to know when you're wrong. And that has been the challenge throughout this pandemic, with myths that have come, that have said that this virus was created by government labs and the government is using it to kill people, to the fact that the vaccine was rushed. And typically when I hear somebody say the vaccine was rushed, it gives the appearance that steps were skipped. And no steps were skipped for making the vaccine. Did it happen fast? It absolutely happened super fast. So I can talk about that some more, that this vaccine will edit your DNA. But as I showed you, there's no way that your mRNA could edit your DNA. The mRNA is in the cytoplasm, your DNA is in the nucleus, right? mRNA and DNA are completely different languages, right? They don't integrate. RNA doesn't go back into the nucleus and integrate into RNA. So that's also something we can talk about, that the vaccine causes infertility in men and women, and sterility in men. Just not the case. When the vaccines were given out, it wasn't purposely given to pregnant women, but I think something like 23 women who got the vaccine became pregnant during the clinical trial. Okay, so there you go. That the vaccine caused irreversible adverse events. This is a vaccine, it's a medication. It will cause adverse events, everything from headaches to fever to pain at the injection site, but these are all reversible. Even for those people who have anaphylaxis or this whole body allergic response, these are reversible events. That vitamin D and other supplements and plant-based supplements can prevent or even treat COVID-19. There is absolutely no data that suggests that. And when you get it, you can show it to me. That the government and Bill Gates have conspired to place microchips in the vaccines to track us. Absolutely not true. If the government needs to track you, all they need to do is to tap on the device that you actually are using to connect right now, right? I mean, it would be silly for them to waste time trying to put microchips into multi-dose vaccines. Remember, each of these vials contain enough for either 5 or 10 doses of the vaccine. How would you ensure that microchips are in every dose of less than 0.5 ml that you're putting into a dose? And that wearing masks will lead to buildup of carbon dioxide that can be bad for your health. Remember, carbon dioxide is a gas. It diffuses very readily through whatever mask you're wearing right now. So that also is a myth. Here's why the vaccines were created so quickly. The typical process that we go through for making a vaccine is the process of figuring out which protein is going to be immunogenic, is going to stimulate a good immune response. developing a technology to make that protein, either through using other bacteria or some factory means, then purifying it, make sure that no toxic substances are in it, then getting enough, figuring out how much of it will work, and then now trying to mass produce that, put it in somebody to make the vaccine. You didn't have to do that with the mRNA technology because most of it was done on a computer, right? The second reason is because the technology, the mRNA technology, was literally ripe for the taking. It had been in development for nearly 20 years, and it was just right at that time. They got a head start of everybody else. The other thing that they did is that they had overlap of phase 1 and 2 trials. Typically, in a general time when you don't have a terrible pandemic, you would complete your phase 1 trial, then move to phase 2 trial, then move to phase 3 trial. What they got authorization from the FDA to overlap trials. So as phase 1 started looking good, they started phase 2. In other words, more people in the trial. Once phase 2 started looking good, they started phase 3. They overlapped. And then at the time when they start phase 3, they actually started mass producing the vaccine doses. You would never do this under normal circumstances because no company, especially small companies like BioNTech and Moderna, would have the money to do that. Because what if the trial fails in phase 3? Like many other trials, I show you that 4 vaccines have failed so far, right? What if it fails when you have already mass produced so much? Well, they didn't care because they were getting federal money to do this, right? The government was bankrolling, especially the Moderna vaccine. And the third, or fourth thing now that makes this vaccine roll out so quick is because the pandemic is so bad. There's so many people getting infected that if you vaccinate people, you don't have to wait very long for the data because so many people in the population have gotten infected. So those are some of the reasons why this trial actually worked. So just to show you how myths can spread, and this is going to be my last slide here. Somebody sent me this video of this person claiming to be a nurse who got the vaccine, and she said she got Bell's palsy. I want to play it for you.
Hi, I am a registered nurse in Nashville, Tennessee, and my name is Kalila Mitchell. I'm reaching out to everyone about the COVID-19 vaccination. I recently took the COVID-19 vaccination. After the shot, I felt fine, but within 3 days, I went to the doctor because I had problems with my face. The whole left side of my face, actually. I have Bell palsy now. And as you can see, I can't smile. I'm trying to smile. It's kind of embarrassing for me. But I just want everyone to know that I think this vaccination is the worst thing ever. And I would not give this to anybody, even my worst enemy. Please, America, they do not care about us. Do not take this vaccination.
So, very emotional, right? I mean, I saw this and almost immediately my heart was breaking for this young woman, but then there were certain things that didn't add up for me. She keeps saying this vaccination is a vaccine. People in the medical field typically don't say I got this COVID-19 vaccination. Vaccination is a process. The vaccine is a product. And then she says Bell palsy instead of Bell's palsy. So, but I waited and then went and fact checked it. And so did many other people. And so what they came up with is that she said she identified herself as a nurse from Tennessee and gave her name. Well, it turns out that there's no nurse in Tennessee by that name. Number two, they went and checked the entire other 49 states. There is absolutely no nurse with that name and or description. So the question is, why would somebody put this out if it's not true? You see, the thing is that you have to be mindful of is that when you're seeing these stories, is how clever the anti-vax movement is. The vaccine rollout in this country was started when they actually gave the vaccine to an African-American nurse as the first person. The response by the anti-vax movement was to put a video like this out to show an African-American young woman claiming to be a nurse who got the vaccine and got one of the worst possible side effects you could possibly get from a vaccine, and give the impression that if you get such a side effect, it will be permanent. So you have to be very careful about what you listen to and what you read, because people will use it to try and get you. And I'll stop now and take some of your questions that you might have. Dr. Honore or Sister Honore, if there are questions, I'm not sure how the question will work.
Yes.
Go ahead.
So we want to basically apologize for some of the technical issues that some people are saying, but we've reached the time now for questions. And in looking at the questions, one talks about side effects. Can you guarantee that there are no long-term side effects? So there are a number of questions dealing about safety and side effect of the vaccine. Dr. Webley, would you be able to address those, please?
Yeah. So again, this is a medication. There's no way anybody can guarantee that you won't have side effects. Where long-term side effects from vaccines are concerned, that's highly unusual. In fact, when you looked at the FDA data, the FDA went back and ran some statistics for the past 50 years, looking at all the vaccines that they have approved. And they realized that the average time for side effects to be reported is within 2 months of people having gotten that vaccine. And that's why neither both the Moderna and the Pfizer vaccines, they waited for 2 months before they applied for emergency use authorization. After that, it will be highly unusual to have any side effects from a vaccine, especially a vaccine of this type. Remember, this vaccine doesn't contain the virus. This vaccine doesn't have preservatives. This vaccine doesn't have a lot of the other things that some traditional vaccines might have. All that it has really is this piece of message, or these pieces of messages that tells it, the body, to create proteins from this, so that now the immune system, once the immune system sees these proteins, it realizes that they're foreign, and it makes antibodies against it. It has memory cells and plasma cells and all those created. So once the message has been read, so what these two companies did is that they caused each molecule of messenger RNA to produce multiple copies of the proteins, probably up to 100 copies of the proteins from one message. But once that message has been read and all the proteins have been made, the messenger RNA gets destroyed. All right, messenger RNA, like anything else, any other product in the body, is susceptible to enzymes in the body destroying it. So it gets destroyed. It doesn't sit around for a long time. In fact, within hours of it being used up, it will be destroyed. It doesn't self-renew. So this is not sitting around in your body for a long time. Yes, Sister Evelyn, I see you waving your hand. Unmute.
Well, while we're waiting, there's another question here. How long does the immunity last after the vaccine?
Good question. I wish we knew that, but we don't know that yet, since the vaccines have just started being deployed widely, broadly. But remember that phase 1 of these clinical trials started over the summer of 2020. You know, you're talking about May, June. people were going through phase 1, and those phase 1 people still have immunity. So we know that for at least that long, the immunity will last. But at this stage, it's too early for us to tell how long immunity will last against these vaccines. The hope is that it'll last for a long time, and that we'll be protected for multiple years with these vaccines.
Okay, we're just getting bombarded with questions, so I'll just keep them coming. You said that the RNA vaccine is not a new science, but it is new being used and tested on humans.
So it's not new, and it's not the first time it's being tested on humans. Again, as I showed in the papers that I showed in 2017, 2019, it was already being tested in humans for different types of vaccines and different types of products, including everything from cancer to diabetes. So this whole mRNA technology is something that has been around for quite a while. A couple of scientists at the University in Pennsylvania actually figured out this technology. And in 2005, they figured out how to actually prevent the mRNA. Because you see, before, when they took the naked mRNA and they put it in, it would just get destroyed. And so they had to figure out a way to stabilize it and a way to protect it and to get it to the place where it could actually stay and be translated. And so the technology was in development over a long period of time. In other words, I'm saying that this is not a technology that was developed once COVID-19 came about. Right? It wasn't developed because of COVID-19. It was there before COVID-19 came around.
Okay. And another person is asking, it is said that the vaccine contained parts from human fetuses. What can you say about that?
That is absolutely not true. Again, as I told you, these vaccines, all they have in them is that they have the messenger RNA. They have some lipid particles. These are lipids, fats. Lipids are fats that are manufactured so that they can encapsulate the vaccine. They have polyethylene glycol, which is an agent that we use in the lab. It helps to make membranes fuse. So they probably keep the fat membrane together and help those fat particles to fuse to our cells. And then it has salt. It has normal saline, the 0.9% saline that you get when you go to the doctors. And they're giving you fluids, and that we use in all research labs all around. So that now is phosphate buffered saline. So it has different types of salts, potassium and some other types of salts, sodium chloride, of course. And then sucrose. Sucrose is a sugar that is put in it as an ingredient as well. That's all that's in these two vaccines. Now, there are vaccines that are being developed. So, for example, the Astrazeneca vaccine is on a completely different platform. The Astrazeneca vaccine is a viral vector vaccine. In other words, what they're doing, they're using a virus that doesn't cause disease to deliver the genetic code for this SARS-CoV-2 spike protein, right? So what they do is that they use this virus, adenoviruses, which we grow in our labs all the time, but they need a human or animal cell line in order to grow. And so what they do is that they grow them up in fetal cells, aborted fetal cells, and these fetal cells were aborted in 1963, to be precise. And what people don't realize is that cell lines are kept for hundreds of years if you actually have the capacity. In -80 freezers, we have tons and tons of cell lines in our -80 freezer. At any time we're ready to grow them up, we just go and thaw them and put them in an incubator at 37 degrees and they grow. So those are the cells that they grow these adenoviruses, these Trojan horse viruses in. Then they purify them out of the human cells. And that's what they give to the person. So that's the AstraZeneca and other vaccines that use these viral vectors. The vector itself, the virus itself, has to be grown in something. Can't just leave it growing on its own. And so essentially then there are no human or animal products in the Pfizer and Moderna vaccines.
Thank you. Another question here. These vaccines were developed so fast. How can we trust them as being safe when the manufacturers have protected themselves from liability?
So this is another common misunderstanding. And people say this because the anti-vax movement keeps saying it. Vaccine companies have never had liability for anything happening from a vaccine. The government has a system set up where you can go and report adverse events from vaccines and get compensated that way. No vaccine company in the history of making vaccine has ever accepted liability for any risk coming from the vaccines. Again, what I explained to you before, towards the end of the presentation, is that the vaccines, when we say that the vaccines were developed quickly, and based on the end of the question, how can we, say that they're not going to be dangerous? The idea is that somehow there's some fundamental steps that were skipped in the production of the vaccine, and that did not happen. These vaccines underwent the same scientific rigor that any vaccine would undergo. And they came out on the other side being safe and efficacious. So it is not true that these vaccines were rushed in the sense that, some steps were skipped. So here's the thing. Let's say that you went home and found your loved one fallen over in your living room. The first thing you do is that you get on the phone and you call 911, right? You don't expect that the ambulance is going to sit around in traffic, obeying every traffic sign, and then get to your loved one. You want them there as fast as you can, right? And so there are laws and rules in place where an ambulance can put on its sirens, every vehicle pull over, you give it space. When it reaches a red light, it cautiously goes through that red light so that it can get to you in the shortest time possible. That is what these emergency uses are. There are already rules in place. Not because this ambulance has a siren on mean that it can do anything it wants. They're trained. The driver of the ambulance is trained into how to do this so that they can safely navigate traffic and get to you in as fast a time as possible. That's what's happened with these vaccines. These overlapping of phases is the ambulance with the sirens on, getting the product out as fast as you can. And that's why these vaccines are under emergency use authorization and not full authorization, which means that every batch they have to look at closely to make sure that it actually has the same product that the initial batch that was authorized actually has.
Thank you. From Myers to everyone, the mRNA vaccines methodology of using our cells' ribosomes to make protein sounds similar to the way coronavirus works in overpowering a cell to make copies of itself. How much spike protein can the vaccine cause the body to make before mRNA is destroyed?
Okay, good question. So you're right. It's the same thing that the virus does, and any virus or any microbe that is an obligate intracellular. It has to co-opt or hijack the body's systems or factory, protein-making factory, to make its own protein so that it can be productive. As to how many copies can be made, one copy, one mRNA copy that is put in for the BioNTech vaccine specifically can actually cause 100 copies to be made, because for the person who's asking seems to know a little bit about the science. So what they did is that they put a poly A tail on the end of the sequence, and they put 100 A's on it, which means that it can make 100 copies if it does it efficiently. So you're talking about, for the dose of the vaccine with about 30 micrograms of mRNA, probably in the order of about 11 to 15 billion copies of protein, of spike protein would be produced. And that's decent amount of protein, but not an overkill. So now with the shortages that we're seeing, that's a concern as to whether or not you'll be able to get the second shot in the time period that is necessary. You should be able to get the second shot, period. But within that time frame of 21 days for the Pfizer, 28 days for the Moderna. I don't know. I don't know what will happen. My thought is that the authorization that was given by the FDA is for what we saw in the clinical trial. The clinical trial had these vaccines, these shots, these booster shot after that period of time that I just specified. Nobody really knows what happens if you go beyond that. Now, from an immunologic perspective, in theory, nothing big should happen. You would still have memory cells there. You should still have cells that can ramp up the immune system. The question is whether or not you'll get to that exact 95%, or it will be 92% or 91% based on the amount of time. Remember that timing is important, because what you're doing is that you're giving your body enough time to maximize the production of antibodies and maximizing the production of memory cells, which are really important in that booster, because what you're doing is that you're now increasing the levels of circulating antibody proteins that can neutralize the virus, but you're also neutralizing your pool of memory cells, and so that you can have those over long term. Now, as to how long this protection will last, we just don't know how long this protection will last from this from these vaccines. We just have to wait and see.
Okay.
Then the next question deals with the, let's see, there are a number of questions and I'm trying to kind of loop them all together. So the next question is, can you comment on the efficacy of the current vaccine on the new strains?
Yeah, good question. So these new strains of the virus, you have this B.1.1.7 strain out of the UK. And then you have that 501 strain out of South Africa. There are other strains coming out of Brazil and other places, and that's always a concern. But this is what viruses do. They're constantly mutating. And although this is an mRNA virus, it actually has a proofreading system, believe it or not. So it has a system like your spell check on your computer that catches its mistakes and correct them. That is why we don't have more mutations than we have now. If it didn't have that, it would be like an HIV, where it makes mistakes randomly and you can't really catch up with them. The more you have the virus spreading in the community, the more chances you have of making these mutations. The good news so far is that the Astrazeneca the Pfizer vaccine, they've gone back and checked and found out that a paper just came out that showed that it offers pretty much full protection against the UK strain. However, when they looked at they're now looking at the South African strain, and that is not offering full protection. However, the level of protection that it offers is still enough to protect you from having serious disease. Because remember, you're talking about a 95% protection from these. That has never happened before for anything that we've seen, probably beyond measles. So, you know, at the time when these vaccines were being developed, the thought was that if we got anything that had 50% protection, we would gladly take it, right? But these vaccines now are offering very high level of protection, which means that they have some buffer, that even if there's a reduction in protection, there will not be this total avoidance of antibodies that are being produced. And so that's our hope. However, even if it's the case that the new strains have evaded the vaccine, because of the ease with which you can redo this platform, they can just go in and redo the mRNA and make a new mRNA vaccine that we would get as a booster that could protect us. Now, the other part of the first question that I forgot to answer is whether or not you would have to get a booster each year. The hope is not. The reason you have to get a flu shot each year is because you're getting different strains of the flu virus. That's not the same case with this new virus, with this coronavirus. Again, it has a proofreading system, not making as many mistakes. The idea is that one vaccine should protect you over multiple years until the amount of antibodies have waned. And again, we don't know how long that is. We will find that out as time passes.
Okay. And we just want to let our listeners know that we did have technical difficulties. This program will be replayed in its entirety on our social media pages so that everybody can hear what took place today. Let's go now to another question. I heard of people who took the vaccine and came down with the virus. Any comments on that?
Yes. So if you get the vaccine, let's say I got the vaccine today, and got exposed to the virus a week from now, I would still get infected. Because remember that 2 week that I told you about? It takes that much time for you to produce protective antibodies. But even so, even after getting both doses of the vaccine doesn't prevent infection. The vaccine prevents disease. You have to get infected for the vaccine to protect you. That's true for every vaccine. So there's no vaccine that puts up a shield around you that says no virus can enter this house, right? That's not what vaccines do. What vaccines do is that they prime your immune system so that when you actually encounter the virus, you probably don't even know that it was there because the immune system just takes it out, right? And neutralizes it and prevents it from causing disease. So that's essentially what the vaccine is designed to do. So for people who have gotten the first shot of the vaccine and haven't yet gotten the second shot, they only have about a 50-something to 60% protection. For these Astrazeneca and Moderna vaccines, after you've gotten the second shot, under the best case scenario, you have a 95% protection. In other words, the probability of you developing severe disease to the point where you're sick, have to be in bed, or have to go to the hospital to get treated is less than 5% of people in 100, right? So 5 in 100 might need that. But overall, that's what vaccines do. They prevent you from having severe disease to the point where you're going to have long-term sequelae. And I showed you like the COVID tongue and the loss of taste and the loss of smell and the nausea and the difficulty breathing and the intestinal problems and the kidney problems that this virus is able to cause. The vaccine will be able to protect you from all of those.
Okay. Okay. I just lost the chat.
Somebody's sharing their screen. Somebody's sharing their screen. You have to stop screen share.
Whoever is sharing a screen, please stop. Thank you.
Screen share is still there. It's Lynette. You're sharing your screen.
Okay.
You should be able to go up to the top and stop the screen share, Dr. Honore, if you have full.
All right. Let me get to another question.
One question is, could you speak to the major difference between the Pfizer and the Moderna vaccine?
Yeah, I mean, they used a little variation in the code for the way they developed their vaccines. They used a different combination of lipids. But really, the major difference is that the Pfizer vaccine requires ultra cold temperature. It requires you to have the vaccine stored at minus 80 degrees prior to you opening it up and going in. And once you go in, you have a limited amount of time before you have to use up all the doses. For the Moderna vaccine, it's stored typically at a minus 20, and after you open it, you have up to 30 days that you can use it. As one of them, the Moderna, each vial has 10 doses in there. The Pfizer has 5 doses, but overall they offer similar levels of protection. You know, you're talking about 94 point something to 95% protection. So they're very, very similar in the way they're made. They're both mRNA vaccines. That's why they have similar efficacy. So they're not huge differences.
We're going to allow some participants to ask questions directly. So we're going to open up. You are allowed to unmute yourselves now if you want to ask a specific question while we wait. Are there any people, any category of people who don't need to take the vaccine? Okay.
Okay, thank you. This vaccine was not tested. None of these vaccines were tested on anybody below age 16. So anybody below age 16, so for the Pfizer vaccine was tested on 16 years up. The Moderna vaccine was tested on 18-year-olds up. So anybody under those ages are not eligible to take the vaccine. The vaccine was not tested on pregnant women. And so if a person is pregnant, you wouldn't be given the vaccine. Although I have a friend who is pregnant and opted to take the vaccine. Other groups could be people with severe allergic reactions, people who, especially when they get injected, have severe allergic reactions. Those individuals might not want to take the vaccine, or they'll be advised to talk with their doctors and take the vaccine under observation. In other words, in a medical facility where there's an EpiPen available, just in case you have an anaphylactic shock. Outside of that, there probably are people who have a lowered immune system. People who are probably undergoing chemotherapy, for example, that might have wiped out your immune system. You don't want to take the vaccine. Not that the vaccine is going to do you anything, but that you might not be able to make the type of response that you need for protection. This has not been put out there, but my impression is that this vaccine should not be given to individuals who are so sick that they have been given a short time to live. Because it's very likely that the fragile state in which they are, when the vaccine is given, it will ramp up inflammation that could actually be detrimental to the health of these individuals. They're people who can barely move. Any sort of fever would be damaging to them. And when you get a vaccine like this, any vaccine that's going to work properly, the first thing that's going to happen is that your immune system is going to ramp up. When your immune system ramps up, it's like having a V8 engine that you're revving, at all the time. It's going to get heated.
Hello?
Yes, go ahead.
Yes, thank you for the presentation, Doctor. I was just wondering that if it is possible, as you've been presenting the information, that a completely safe vaccine could be produced in 10 months, then why has the protocol been that nothing has been released as far as medication or vaccine development? Nothing actually has been released to the public after several years of observation and testing. I guess that's the part that doesn't seem to connect for me. These protocols seem to have been put in place for a reason and for observation, but here it seems like, okay, 10 months, we have everything we need. You've mentioned the mRNA technology has been out there for 10 plus years, but nothing has actually been distributed or made for public consumption. It's only been stuck in the phases, which was part of the original protocols put in place. So why the disconnect? Why is it all of a sudden now that it's found that it's okay or it's safe to be placing the vaccine out for public use in such a short amount of time without really any knowledge? I mean, despite what you were stating, which I can understand, they're not seeing any side effects, but at the same time, historically, they haven't put out anything at this large a scale that's been made in this specific form to actually observe if there are any long-term side effects. You're dealing with manipulating genetic information. And
all right, can we allow him to answer? Go ahead.
I'm sorry, thank you.
Yeah, I get the question and I get the passion that it comes with as well. However, science is science, right? The science bears this out. And so what you're talking about apples and oranges. The original vaccine platforms that have been used required that amount of time. So it wasn't that phase 1 was taking 3 years because they were observing for long-term side effects. That's not the purpose of the phases. The reason the phases were taking that long was because they just couldn't get them to go any faster. Right? The reason you're waiting for phase 2 and phase 3 to give you data is that most of the diseases that you were testing on weren't around in the level that SARS-CoV-2 has caused disease. In other words, they were able to turn around these vaccines quickly because they were able to get data from their clinical trials in a very short time. When you're having 200,000 people being infected each day, it doesn't take a long time before people in your different phases of your clinical trial to get infected with the virus, which is exactly what you want. So let me explain to you what is going on in clinical trials. In the phase of a clinical trial, what you're trying to do, you're looking to see when I Bristol 5, whatever that is, Bristol 5, you mute.
I have to mute everyone again because it's just not going to work if all the mics are open. Go ahead, doctor.
Yeah, so, in your phase 1 clinical trial, let me just explain clinical trials a little bit to see if I can answer this question. Your phase 1 clinical trial is done on a handful of people, 40 to 50 people at most. And what you're doing in phase 1 clinical trial, you're doing a couple things. You're not even testing to see if the vaccine works in your phase 1 clinical trial. What you're doing is that you're trying to see if it is safe. And so you're doing what we call dose escalation studies. In other words, you're giving different groups of people different doses of the vaccine, and you're seeing which group has side effects or adverse events that are outside of what you would feel is acceptable based on prior vaccine studies. You're hoping that you will see some data that shows efficacy, but that's not the primary goal. Moreover, the sample size is too small to determine that. So once you've done with phase 1 clinical trial, you go into phase 2, where you get to do hundreds of people. And now you can actually have the statistics to look at efficacy. There's nothing that says you have to wait X amount of time for a phase 1 or a phase 2. The phase is done when you have enough people in the study who have had the shot over a period of time. Phase 3 clinical trial now is where the rubber hits the road. This is a double-blind, case-control study where you give one group of people a placebo, maybe you give them some sugar and water or some saline or something like that, and you give the next group the vaccine. So you enroll a lot of people, tens of thousands of people. In this case, for both the Pfizer and the Astrazeneca and the Moderna vaccine, over 30,000 people. I think the Pfizer had 40-something thousand people were enrolled in the study. Half of them were randomly given a placebo. The next half was given the vaccine. Neither the patient nor the vaccine company nor the person giving the vaccine knows who is getting vaccine versus